Hi, my name is Tom, and today my group and I will be presenting our project on the effects of short-term daily Coke Zero consumption on the gut microbiome. We focus on non-nutritive sweeteners. Firstly, let me define on what non-nutritive sweeteners refer to. They simply refer to any additive that can be added to foods to increase their sweetness while providing no or very little caloric value. Some common examples include aspartame, sucralose, and stevia. In our project, we'll be focusing most of our discussion on sucralose. And you may ask why. The main reason is that sucralose cannot be digested by the host and reaches our colon relatively unchanged. This allows for the largest avenue of interaction with our gut microbiome, and studies show that they can possibly function as an antimicrobial or a potential carbon source. You may also ask why the use of sucralose is so popular. And these can be attributed to two very important reasons, one of them being reduced calorie intake, as well as preservation of food choices for diabetic patients, hence improving their quality of life. However, they are not all perfect, and many studies have shown that daily sucralose consumption can have an effect on our gut microbiome. Here are two of the many studies, and they seem to contradict each other. The paper on the left shows that there are no significant changes on the phylum and genera level. However, the paper on the right shows a significant change on the genus level as well as a microbial metabolic level.
Many of these studies that analyze the impact of sucralose on the gut microbiome are human randomized controlled trials. In these studies, fecal samples were collected just as we did, and in most of these studies, as expected, the Firmicutes and Bacteroidetes phyla were the most dominant before and after intervention in placebo and the treatment group. Usually, the microbiome composition tended to remain stable in both interventions, and most subjects displayed only modest variations. Not all studies found significant differences between participants who consumed sucralose and those who didn't. A randomized control trial was designed to determine how different sucralose regimens affect the human gut microbiome. For the two short-term exposures, none of them caused significant effect on microbial composition. The third arm extended the exposure time to 10 weeks, and this prolonged intake lower than the ADE of sucralose that led to clear signs of dysbiosis. Specifically, levels of lactobacillus rose significantly, while the population of Bacteriobacillus, Lactobacillus, Acidophilus declined. And these results suggest that to have a significant impact on the gut microbiota, the sucralose intake has to be maintained for a long period of time. And some researchers observed also an increase in blood lactobacillus, a bacterium linked to improved blood sugar control, and a decrease in lactobacillus. For our study, we wanted to find out whether short-term and daily consumption of sucralose, specifically through the Coke Zero, could influence the composition and diversity of our rectal microbiome, and can these changes be associated with dietary and lifestyle factors?
For our intervention, we chose Coke Zero because it contains sucralose, the non-nutritive sweetener we wanted to study, along with acesulfame K, another common sweetener. Using a commercial drink keeps our experiment realistic and convenient. It is widely available, portable, and fits easily into our daily routines. Since many people drink Coke Zero regularly, it also lets us examine whether everyday levels of sucralose intake can alter the gut microbiome. Before starting, we included a 1-week washout period to let participants' microbiomes return to baseline. Although literature suggests 4 weeks would be ideal, we shortened it for practicality. This means residual dietary effects might still influence our results. During the intervention week, participants drank 2 cans of Coke Zero per day, 1 at lunch and 1 at dinner, roughly 10 to 17 milligrams of sucralose daily. Since acesulfame K is absorbed and excreted through urine, it likely doesn't interact directly with the gut microbiota. This is far below regulatory limits, which range from 5 mg per kg to 15 mg per kg, according to the FDA and EU respectively. Typical consumers take in only 1 to 3% of those limits, though heavy consumers may reach about 6 to 15%. Hence, our design reflects realistic, safe, and ecologically valid consumption.
Next up, I'll be talking about our group's study design. Our experiment will be conducted in 2 phases, the washout period as well as the intervention period, both at 1 week each. 2 sets of samples will be taken, once before the intervention and once after. Let me explain more about our study design. The washout period's intended purpose is to reset and stabilize the gut's microbiome back to the basal level for all the participants. During this period, as well as the intervention period, a strict diet plan is followed where consumption of additional NSS such as sucralose and any supplementary pre- and probiotics are stopped. Participants also followed the Health Promotion Board's diet guideline, abiding by the macronutrients and fiber intake requirements to ensure that we reduce as many confounding factors as possible. Lastly, during the intervention period, we consume 1 can of Coke Zero during lunch and dinner daily for 1 week. As said earlier, we collected 2 sets of samples for our experiment. Our collection method is via a rectal swab. A sterile swab is inserted into the anus 2 to 3 cm deep. The swab is then twisted a few times to ensure sufficient sample is collected from the mucosal walls of the rectum. Lastly, triplicates of each swab were also taken to ensure higher count of microbes collected for our pre- and post-intervention analysis.
Thank you, Shaz. I will now talk about the pros and cons of our sample collection method. For our study, we used triplicate rectal swabs to collect samples before and after the 1-week sucralose intervention. This method is simple, quick, and minimally invasive, which makes it easy for participants like us to follow. It also captures mucosal-associated microbes that tend to be more stable and functionally relevant to health compared to those found in stool. However, there are some drawbacks. The risk of contamination is higher since samples were collected in toilets, and the technique can vary among participants, which affects consistency. Rectal swabs also only capture a small portion of the gut microbiome and produce lower biomass, which can and did reduce sensitivity in downstream analysis. To reduce variability, we controlled for diet using daily food diaries that track macronutrients and fiber intakes according to the HPB guidelines with a 5% allowance. We also included a 1-week washout period so that we can stabilize everyone's baseline microbiome. Here's an example of our macronutrient tracking. These steps are important because diet strongly influences microbial composition, so standardizing it helps to ensure that any observed changes are due to sucralose rather than diet differences. However, some confounding factors still remain. These include inconsistent diet, varying dosage times, and lifestyle differences such as sleep, stress, and physical activity can all influence microbial responses.
Our study design, while practical, had some limitations affecting results interpretation. First, diet and lifestyle variation. Although we track macronutrients and fiber intake per HPV guidelines, day-to-day differences in sleep, stress levels, and physical activity can influence gut microbes independently of the intervention. Next, Coke Zero isn't a pure sucralose source. It also contains azosulfame K, caffeine, phosphoric acid, etc., which can independently influence gut microbes. Any observed microbial changes can't be attributed solely to sucralose. Also, data on sucralose content in Coke Zero is limited. We estimated based on similar drinks like Pepsi Zero, however this may not reflect the true dose, making it hard to judge whether exposure was sufficient for microbial effects. Biological variability between participants also matters. Differences in gut transit time, baseline microbiome and metabolism mean the same dose can affect people differently. Our small sample size limits statistical power and generalizability. With few participants, hundreds of microbial taxa, and high natural variability, random variation may overshadow true effects, making it harder to detect real trends or adjust for confounders, increasing the risk of overfitting. Finally, the short 1-week intervention likely cannot capture stable microbial adaptation. Slow-growing bacterial communities need weeks to establish new equilibria, so our findings may only reflect transient changes rather than the true ecological shifts. Typical studies use around 10 weeks instead.
Now I'll move on to discuss the significance of our study's limitation. Firstly, the presence of confounding factors make it difficult to isolate the true effect of Coke Zero on the gut microbiome. Ingredients like azosulfame K and caffeine could either mask or mimic the microbial changes that we might. Secondly, the unclear sucralose levels in the beverage limits our ability to compare our results with established dose-response data from previous studies. Altogether, these limitations reduce the internal validity of our findings, making it harder to draw causal conclusions about sucralose's impact on microbiome. On top of dealing with the limitations of our current study, such as using and quantifying the pure sucralose solution instead of commercial beverages, and then measuring the sucralose concentration in both beverage and participants' stool sample, increasing the sample size and extending the intervention period to improve generalizability, controlling diet and lifestyle plan, and adding a control group that consumes non-NNS beverages, we could also collect multiple time point measurements, which will allow us to actually track the both short-term and long-term microbial shifts, helping us review whether participants adapt to or recover after exposure and clarify how long any effect persists. And integrating targeted metabolomic, which will also strengthen our analysis. It links microbial metabolites to functional outcomes and enable multi-omic integration with metagenomic data. We could also do dose response studies to establish causal and identify threshold on non-effect levels and understand individual sensitivity to varying doses. And with that brings us to the end of our presentation.